3, 2, 1.
I think the most exciting part was watching the liftoff of our flies.
As Falcon and Dragon take flight to the International Space Station.
It was so cool. Made all my hair stand out on end. Right now, the plan NASA has is to have a colony on the moon for people to live and work in. If we're going to establish a colony on the moon, or if we're going to have people mining asteroids, then we definitely need to have a better understanding of how microgravity affects all of the different organ systems in the body. Roughly 75% of disease-causing genes in humans are found in the fruit fly. The human heart starts out as a linear tube embryologically and then changes shape, twists to form the four-chamber heart that everyone is familiar with. The fruit fly heart starts out as a tube as well. It also has 4 chambers. It's just that it doesn't ever twist into a more complex organ. In addition, it has a number of very important proteins that are critical for human heart function. So inside this box, we were able to put 15 vials of parent flies, and they laid eggs that then hatched and became adults aboard the ISS, spent an entire month, which for a fruit fly is basically half of its life. And then we got this box back, opened it up, and got approximately 400 flies, adult flies, that were born on the ISS and came back in this box. We did climbing assays to see how their quote-unquote skeletal muscle was working. We collected their hearts. So basically we dissect the flies open. We take high-speed movies of their heart beating in order to be able to quantify how the heart works. And then we remove the hearts and preserve them for subsequent genetic analysis. Visually, they didn't seem to be quite as contractile as their ground control counterparts. They had less ability to squeeze the blood through the heart, and were able to show definitively that their hearts just weren't as strong, if you will, as their ground control siblings. And we also were able to do a number of morphological studies that showed us they had changes in the fibrotic structure around the heart.
That was quite a surprise to see that, in fact, which makes us think that there are things that are happening that are not necessarily expected.
We have come up with some interesting new genes that haven't been described in heart function before that we're pursuing, and so these are potentially related to the remodeling of the heart that occurred in microgravity. And similar kinds of remodeling occurs in human patients when they're confined to bed for extended periods of time or aren't as mobile as one should be. And so if we can identify some of those important genes that may be mediating the remodeling response, then that's of benefit to humans on Earth, as well as to become a target for countermeasures that can be given to astronauts who are going to be spending perhaps 1.5 years in microgravity on their way to Mars.
Ideally, we would like to be able to have more flights to test these kind of hypotheses of what components are the most critical ones.